Hi everyone, my name is Adele, and my internship was with Shadow Lake Nature Reserve on how sphagnum and feather moss species impact bog ecosystem health. For context, my work was in the peat bog ecosystem at Shadow, which is a unique ecosystem that forms over thousands of years when dead vegetation accumulates faster than it can decompose and forms peat soil. Peatlands around the globe are becoming increasingly rare due to degradation and peat extraction. Sphagnum mosses are the keystone species that release acids contributing to peat accumulation and make the environment acidic and anoxic. However, there's a lack of information on the identification methods and distribution of sphagnum moss. Therefore, this project investigates how the abundance and spatial distributions of sphagnum and feather mosses reflect ecosystem health. So this led me to ask, how does the abundance and spatial distribution of sphagnum and feather mosses differ across the peat bog, and what does this reveal about ecosystem health? To answer this question, I first created an identification guide of 29 sphagnum species native to Washington, and then collected data from 15 quadrat sampling plots from each year, calculating percentage coverage of all species, and then analyzed the relationship to microhabitat variables. Image 1 on the left shows me performing this data collection, and image 2 shows a zoomed-in photo with Sphagnum gergensonii at the top and red stem feather moss at the bottom. In our data collection, I was very focused on the identification of sphagnum, as it's typically very challenging and species have microscopic differences, while feather mosses are generally a lot easier to identify. First results show information about the entire plant population at Shadow. In 2025, we identified 19 species, and in 2026, we identified 27. This increase is likely from improved species identification over the year. Sphagnum was identified at 2 sampling plots in 2025, and in 2026 was identified at 4. In 2026, we were able to identify all the sphagnum in the sampling plots as Sphagnum gergensonii. This was very surprising that the species were all one species, because we know that there are likely many more throughout the bog, although gergensonii is one of the most common species to encounter. Among the 5 feather moss species, the red stem feather moss is the most abundant in our 2026 data collection. Figure 1 shows the cumulative species percent cover from both years, totaling 30 plots, with the 2025 data in red and 2026 in blue. The sphagnum is highlighted by the green star on the graph, and red stem feather moss is highlighted by the orange star. And you can see just how abundant the red stem feather moss is, along with stair-step moss, which is another feather moss. The next result is the statistical analysis of sphagnum. Sphagnum abundance was very low in only 6 out of the total 30 plots. This was very surprising, as sphagnum is the keystone species and would typically have high occurrence. In Table 1, you can see the percent cover of these plots from both years. The standard error of this data indicates low precision, showing that the distribution varies a lot from one sampling plot to the next. This is consistent with having a patchy distribution based on microhabitat preferences. Correlating the sphagnum distribution with sun exposure and hummocks can reveal their microhabitat preference. For context, a hummock is a unique topography feature found in peat bogs, and they're raised mounds of peat that form on the ground. The hollows are the low-lying pits or depressions that surround these hummocks. The sphagnum showed no preference for hollows, and only one plot showed preference for full sun exposure instead of shade. Sphagnum gergensonii is actually one of the only sphagnum species that prefers shade instead of sun. This very low abundance and distribution based on microhabitat variables may suggest that there is ecosystem stress. The next result is feather mosses, which are present in 29 out of the 30 total plots. The coefficient of variation is about 41% for 2025 and 47% for 2026, which measures the relative variance, showing very high occurrence and moderate variability across the plots. Figure 2 shows the feather moss relation to hollow and hummock topography, with their percentage coverage shown on the y-axis. This shows that their distribution has no strong preference for specific topography conditions. The feather moss abundance is very high across the different topography conditions, with ranges of percent cover overlapping. This dominance may suggest that feather mosses are outcompeting sphagnum moss species. These results are significant because they indicate that sphagnum has a habitat preference for shade and hummocks, while feather moss cover varies slightly across the hummock gradient. Sphagnum occurrence is very low and targeted on these variables, while feather occurrence is high and dominant. Sphagnum may be shifting to feather mosses due to drier conditions, increased nutrients, shading, and fires. These findings suggest that sphagnum function as a microhabitat specialist group, while feather mosses are generalists with broad environmental tolerance. Literature suggests that sphagnum at shadow is potentially shifting to feather moss species, which this data seems to support. Since we found sphagnum at such low occurrences, if this is the case, this shift may have already occurred, which would represent a very serious environmental change. This research has contributed to shadow's habitat protection and ecological restoration by addressing peatland degradation. Peatlands are valuable through their high carbon accumulation as global warming trends increase. I would like to thank my site supervisor, Cecilia Monahan, faculty advisor, Benjamin Dittbrenner, and my family and friends for supporting me along this project.